Hello and welcome to my first essay video for exam number 4. Today we're going to be talking about filtration in the kidneys, how the different parts of the kidneys have different roles to play and what that looks like, and just the movement of fluids and solutes within our kidneys. So let's get right into it. So in order to really start explaining things, I need to get down the basics real quick. So we have our kidney right here. You can't see that very well. Let's see if this blue is better. We have a kidney, and within that kidney we have lots of these little tube-looking things called nephrons. These nephrons are where our filtration and reabsorption happen. I'm only going to draw a couple, but in reality there's like thousands, hundreds of them. And the top part of our nephrons is going to be in the cortex, or the cortex, and then the bottom part is going to be in the medulla. If we zoom into one of these nephrons, we'll see that we have a pattern of two different types of nephrons. We'll have one like this, with the kind of a short loop down here. And we'll have another one like this, with a much longer loop. We have about 90% of our nephrons are the short loop types, and 10% is the long loop types. And let's go through the 4 main steps of what our kidneys do for us. So the first step is filtration. That's right, layer there. Second step is absorption. Perfect. Third step is secretion. Fourth step is the excretion. The majority of the water, or the liquid part of our blood that gets filtered through our kidneys, is reabsorbed. And that's a good thing. If that were not the case, then we would get dehydrated even faster than we currently do, which is not good. Let's talk about filtration for a second. Where does filtration take place? Well, it takes place at the heads of these nephrons, the circular parts right here, at the heads. And so if we wanted to zoom up on what that looks like, we'll go ahead and zoom up real quick. I'll erase all of these. We'll zoom into that head. The glomerular head is what it's called. And we're going to have a line of cells. These cells are going to be very close to capillaries. So this is blood here. We're going to have a couple of different pumps or channels that allow different solutes through. So there's actually going to be a bit of a space between these cells. And this is the space where water is able to go through. So water goes through and goes into the blood. And the water drags some solutes with it, namely potassium and calcium. So this is in the descending loop, so that part of the nephron if I were to draw it again, I'll draw it over here. Yeah, you can see it. Messed up that drawing. So these cells that I'm drawing right here is in the descending part. So that little tubular that is going down, descending tubular of the nephron. So we have water pulling some solutes with it, potassium and calcium. And then within these cells, we also have, in the descending loop, these aquaporins that are just letting water permeate these cells quite a bit. And we'll have aquaporins on both sides of the cells, so the water is able to go all the way through into the capillaries and into the blood. We have some channels over here. We have this sodium-potassium pump. And so we have quite a few of these on this side of the cell. So we get this gradient built up of sodium. And that's important because that is what helps pull the water down to this side of the cell. Wherever sodium goes, water will go, and vice versa as well. We also get sodium permeating this capillary wall and getting reabsorbed back into their blood. Right? And along with that, there's another pump that is a little bit unique to this part of the body, where we get sodium following its gradient into this blood and pulling with it some glucose. We also have, as a use for this gradient, sodium channels that flow into the cell and pulling with it lactate. And these are secondary active transports because we're using the gradient, or we're using primary active transport, ATP, in the sodium-potassium pump to build up that gradient. And then we use that gradient to bring in larger molecules, or maybe charged molecules, such as lactate and glucose. So that's what's happening in the descending tubular of our nephrons. Now let's move on to a different part of the nephrons. Okay? Let's go further down and go to our ascending tubular, which is right here. We're still going to have the blood, and those cells look pretty much the same across the board. Kind of this funny, funky looking shape. Now here it gets a little bit interesting because we have an exchange of protons, which means we're dealing with the acidity of the blood. And this is kind of where a lot of our protons or excess protons are filtered through. So as we do this, we're going to have, I don't have my notes with me, help me out with this. We're going to have more sodium gradients being built up over here as well. We got sodium coming out, bringing potassium in, 2 potassium in, 3 sodium out, facilitated or using the ATP as the energy there. We're building up that gradient, so we have big sodium outside of the cell, hardly any inside right now. we're going to use that gradient. We're going to have a channel where we're bringing in sodium, and along with it, we're going to pump out a hydrogen or a proton. So for every sodium that we're pumping in, we're pushing out a hydrogen. And what that's going to do is that's going to lower the pH of our cell here. We're kind of going to have an exchange of CO2 and water. Turning into bicarbonate and protons. And so this is one of the ways that we're able to kind of modulate this equilibrium equation. It's a very common equation when talking about filtration and kidneys and the blood. In fact, it's one of the most important things to understand when it comes to our physiology in medicine. As we are pumping out our hydrogens, we're making this side of the equation smaller. We're making hydrogen smaller because we're getting rid of it from the cell. As we're getting rid of it, Le Chatelier's principle tells us that our reactants will push more towards the products because we still have the same concentration of reactants, but we're lessening the amount of products that we're getting. So through Le Chatelier's principle, we can predict that we're going to have more bicarbonate and protons being made as we are getting rid of that hydrogen through this pump that is bringing in sodium in exchange for pushing out hydrogen. And the reason why we change bicarbonate into CO2 and waters because bicarbonate is not permeable to the first membrane, right? So in order to, we do this so that we pump out hydrogen onto this side. And then outside on this side of the cell, we have bicarbonate still. But because we're pumping it out more of our products, Le Chatelier's principle is telling us, all right, now we have more products than we do reactants, so that the equilibrium will push more towards the reactants. So this bicarbonate, which is charged, so it is unable to permeate this cell, will turn into CO2, which is a neutral molecule, which will be able to permeate this cell. So that's how that we're able to get CO2 to come back this way. And then once it does, it'll turn back into bicarbonate. And that's how we get reabsorption of bicarbonate into our blood. We have to change it into an uncharged or a neutral molecule, being CO2 and water, which is able to flow easily through our cells and get back into our blood. Let's talk about another channel now for a second. We're going to talk about chloride channels. We don't often talk about these types of channels, but when we're talking about the kidneys, we need to make sure that we're keeping track of everything that is going to be important for our survival. Redraw a cell here, just getting a little busy. So again, we're going to use mostly the gradient that we're building up with our sodium-potassium pump, which is our primary channel, building up that gradient of potassium outside of the cell, potassium inside of the cell and sodium outside of the cell. Then we're going to use that same pump again, where we're bringing in sodium on this side, and in exchange for pumping out protons or hydrogen. This buildup of protons will have a buildup of a positive charge just outside of the cell wall there. Once this happens, for every proton that we're pumping out, we're leaving behind a negative charge. There's a separation of charge that takes place within the cell, changing the membrane potential. We have this pump right here that uses this charge to pump out that anion or the negative ion, and we bring in chloride. So this is kind of almost like a third-party channel, because we have our primary channel, which primary active transport, using ATP to build up a sodium gradient. Then we use that sodium gradient to pump out hydrogen protons, which builds up a separation of charge within our membrane. And then that separation of charge is what causes this channel to work, where we're pumping out those negative charges and we're bringing in chlorides into our cell. This is an important process because usually chloride is unable to permeate our cell walls because they're kind of larger atoms that are also very negative, very charged. On this side of the channel, we have this special, or this side of the cell, we have this special channel. We don't really need to know exactly what it's called, but an acronym for it is the CFTR. The CFTR is basically a channel that allows that chloride that we brought into the cell to flow through that cell and back into our blood. And of course, we still have those aquaporins on this part of the nephron that allows water to be absorbed back into the blood. Now, as we're absorbing more and more water as we're going through our nephron, the farther we get through our nephron, the more concentrated our solutes become, because we're absorbing as much water as we can from those nephrons. Once we get to the distal part of our nephron, which starts right about here, we actually no longer have those aquaporins available. We also have this membrane that stitches our cells together, essentially blocking off water from being able to pass between the cells. And so as we go further along our nephron, we become more salty or more concentrated. And hopefully that makes sense as to why. Because we're blocking off any access, or beforehand, we're allowing water to leak through and be absorbed as much as possible because we don't want to lose water. But then once we get far enough, that water has no place to go, and we are left with a very high concentration of solutes within our water. And that concentration, when we get to the bottom of our loop, can get as high as 1,200 milliosmoles. And the reason for that is usually only the long loops, the nephrons with the very long loops, are allowed to get to that high amount of concentration. With the shorter loops, we only get down to about 600 milliosmoles, which is still pretty concentrated. Alright, now how does our body know when to kind of increase our absorption or increase our secretion or excretion? By the way, excretion is another word for just urination, right? You pee everything out at the end, and that's how we get rid of a lot of these things that our body kind of filters out for us. Secretion is talking about when we have solutes that are maybe in the blood, have some solute in the blood, and it gets back into the nephron later on. And that usually happens right about here in the nephron, so in that descending loop. You secrete that. We talked about absorption a lot. That's what the majority of what happens in our kidney is: absorption, reabsorption of solutes and water in order to help keep us at a proper concentration concentration of potassium, sodium, and glucose, and all that good stuff. There are three main pathways that our body uses to kind of regulate this filtration system that we have going on in our kidneys. The first one is called RAAS, RAS. RAS is basically used to create this product called Ang2. And then Ang2 in turn increases our thirst. It also stimulates the sympathetic nervous system to constrict our blood vessels, which increases our blood pressure, which then increases blood flow through the kidney, so more blood is becoming filtered. So that's RAS. Then the second way that our body kind of regulates this is we use something called osmoreceptors. Osmoreceptors are receptors that basically sense the constant amount of a certain solute that is being passed through the blood in the kidney. So, for example, this could be something like a sodium receptor. We kind of have an idea of how much sodium is being absorbed or passing through in our blood, and then we, by turn, are able to tell how much blood is actually coming through the kidney. And so we can have kind of a feedback system where we have our automatic nervous system stimulates us to increase our blood pressure, which increases blood flow, or decrease blood pressure, which decreases blood flow through the kidneys. Finally, we have something called aldosterone. So this increases that compound that we talked about, Ang2, which in turn essentially it increases the amount of blood flow that goes through our kidneys. So there's more than one way to do the same thing, which is why sometimes if we have a drug that is inhibiting one of these systems, it's not completely fatal to us. And we can still reap the benefits of our body regulating this blood flow through our kidneys and therefore the amount of filtration that is happening without really messing with the system. We have kind of a fail-safe that our body builds up for us. Anyways, that was my video for this week. Thanks for watching and see you next time.